Conference recording has started.
That's all right. I better not make a mistake now. Okay, so the E2F family of transcription.
Can I use the interruption to ask, are there any retinoblastomas, the cancer, that would be mediated by mutations in E2F?
There's thought to be amplification of the E2F3 transcription factor in some retinoblastomas. Whether or not that comes without a mutation in RB, probably not. E2F mutations in cancer are pretty rare, though, but there are some here and there. So this is a large family of transcription factors. They're typical in that they contain DNA binding domains, a transactivation domain. One important point is that the retinoblastoma protein binds to the C-terminal region of E2Fs and blocks or masks the transcriptional activation region. Now, I'd like to take a minute and talk about most of my postdoctoral work was on the E2F1 transcription factor, and I'd like to use this to set the stage for the conceptual question that we've been trying to address. Now, in addition to E2F's ability to induce cell cycle entry and expression of DNA replication genes, it carries with it an innate ability to induce cell death. This is through the transcriptional upregulation of pro-apoptotic genes. It can induce the p53 pathway and other pro-apoptotic genes. The question that we've been pondering is, since E2F is upregulated during a normal cell cycle entry, how is this regulated? What signals block this apoptosis during normal cell cycle entry? And if those can be deciphered, does cancer utilize the same mechanisms to suppress this programmed cell death? As, after all, evasion of apoptosis is one of the hallmarks of cancer. And then finally, from a therapeutic standpoint, if cancer does suppress E2F induction of apoptosis, is there a way to modulate that pathway and restore latent E2F apoptotic activity to kill a tumor cell? So a lot of work that I did actually showed the involvement of the PI3 kinase PTEN AKT pathway in suppressing apoptosis here. So PI3K is a major human oncogene. It's a lipid kinase that sustains oncogenic mutations in a variety of cancers, notably breast cancer, for example. PTEN is a frequently mutated, a tumor suppressor gene in human cancer, and it opposes the function of PI3 kinase. So it's a lipid phosphatase. So it's an oversimplification to say that all they do is oppose each other's functions, but they do. PI3K is a lipid kinase, and PTEN removes the lipid kinase or phosphorylation event deposited by PI3K. And mutation of PTEN or activation of PI3 kinase both have this shared feature of activating this AKT serine threonine kinase, which is a well-known pro-survival factor. And one of the things that we were able to show is that activation of AKT interfaces in some way with the E2F1 transcription factor to do a few things. First, it blocks apoptosis, and then later we were able to show that it actually affects the transcriptional output of E2F1 to shut down expression of the pro-apoptotic genes, but without affecting its ability to induce proliferation and expression of these genes. So the real questions that we have then, that the rest of this talk will be about, are really, does this matter in vivo? Because these studies came from plates of cells where we were analyzing these pathways. Does this actually matter in a tumor? And then second, if AKT does suppress E2F apoptotic output in a tumor, what's the mechanism? Because as you may know, AKT functions at the plasma membrane. E2F is a transcription factor that binds to DNA in the nucleus. What connects these two? Yeah?
Before you get further, so the E2F family, is this redundant? Does the same pathway that works for E2F1 work for the other E2Fs?
No. So E2F, it's been fairly controversial because, as you might expect, when you get a large family like this, just knocking out one of them, you run into problems, especially in mice, with compensation from others. There's a few things known about E2F1. Its really in a variety of biological contexts, the only one of these E2Fs that's pro-apoptotic, that's debated, and you can enforce apoptosis by some of the other E2Fs. The other thing that I won't touch on that to me underscores the difference between E2F1 from the others is that E2F1 is remarkably responsive to the DNA damage response. So if you hit cells with DNA damaging agents, you see a dramatic increase in accumulation of certain proteins like P53. E2F follows that same ATM check-2 accumulation, unlike any of the other E2Fs. And how that fits into normal cell cycle passage is really unclear right now. Okay, so we're testing. Really what we want to test here is if we express E2F1, we know that it induces proliferation, but it also induces programmed cell death. The question is, if we co-activate AKT in the same context, does it suppress apoptosis and drive a tumor? We're testing this, and I'll be talking about this in a retinoblastoma mouse model that we developed. But I want to just take a few minutes here and explain, is there any evidence, or what's the thought process behind PI3 kinase activation in human RB? So it's fairly unknown. Really what's unknown is how do retinal cells evade apoptosis en route to cancer development is remarkably poorly understood. But there are a number of reasons to believe that PI3 kinase is activated in some RBs. So epigenetic studies have shown that a Syk kinase, which is a cancer oncogene, is upregulated in some human retinoblastomas. The effect that Syk has on signaling pathways in human RB is completely unknown, but in other contexts, such as diffuse large B-cell lymphomas, it activates PI3 kinase pathways strongly. Second, this microRNA cluster, miR-1792, is amplified in human RB. And again, the effect that this has on RB pathway signaling is unclear. It has not been established, but in other contexts, it strongly activates PI3 kinase and AKT to suppress cell death. And then finally, while rare, oncogenic mutations in PI3 kinase itself have been detected in human retinoblastoma. Okay, so the mouse model that we're testing, the background, other mouse models of mouse retinoblastoma have knocked out RB and P107. I'll give a little bit of explanation to this because it's different than humans. Typically give rise to late onset unilateral tumors in these mice. And again, I mentioned this briefly, but the question, just to reiterate, that we're asking, with high level activation of E2Fs, does P10 deletion, in other words, to activate the AKT pathway, affect E2F apoptosis or tumor onset in this context? And the genes that we're looking at here in this mouse model, first, RB and P10 are essential genes. Germline deletions are embryonic lethal in mice, so we need to use conditional deletions of these with RB lox. The CRE that we're using to drive expression of CRE is retinal specific, so a CHX10 CRE. It expresses in the retinal progenitor cells beginning at embryonic day 10.5, so early on, and all retinal cells are affected by this. P107 is an RB family member that we need to co-mutate because RB deletion in the mouse basically has no effect on tumor onset. One difference between mice and humans is that in mice, P107 is upregulated and thought to be compensating for the loss of RB here, whereas that doesn't happen in human cells. So previous mouse models have co-mutated RB and P107, and the major effect in this context is high-level activation of E2Fs. And then P10, I mentioned, is the lipid phosphatase tumor suppressor that opposes the activity of PI3 kinase and highly activates AKT when active. So we've co-mutated these together to ask this question in murine retina. So the first data slide, we're looking at mice here. The first is control mice, and we're following these out over a 90-day time course, with most of the time points being within 30 days. So And this is a normal retina. I'll show you a close-up of this in the next slide, but it develops normally. All that's expressed in this is the CHX10 Cre. Now, this mice is mutant for RBNP107. We call it a double knockout just to simplify. And we see something very striking even by 15 days. So at 7 days, the retina is developing, but by 15 days, much of the tissue has been lost. And by 90 days, there's a small strand of cells that remains that I'll show you in a close-up in the next slide. Co-mutating P10 in this background, and RBP107 P10, however, has a very striking difference in that early on, we see tumors forming. By 30 days, we see 100% penetrant bilateral retinoblastoma in these mice, and by 90 days, the tumors are huge. So the summary from this is that activation or deletion of P10 in this context drives strong bilateral retinoblastoma formation. Now, this is a close-up just because in the next slide I'll be showing an immunophenotype of some of the more of the details of what we're looking at with the retina. So this is a mouse wild-type retina comparing to a textbook description of the different cell types. And there are 7 cell types that form in the retina. So there are rods and cones, and you can see these in the mouse histology section. Mueller cells, horizontal, bipolar, amacrine ganglion, ganglion cells here. So this is a normal wild-type retina in the mouse at 30 days. So what I'm showing here is an immunophenotype of the 7-day-old mice and looking at just a histology section, H&E, and then a Ki67 is a marker for proliferation, and caspase-3 is a marker for cell death. So, and again, the RBP107, to reiterate from the previous slide, these are the ones where the retina is destroyed by 30 days, whereas the RBP107 P10 mice get bilateral tumors by 30 days. So when we look discretely at proliferation in caspase-3 in this background, we see that there's a little bit of proliferation in caspase-3 that's going on at 7 days. In the RBP107 or E2F-deregulated mice, we see major proliferation still happening across all sections of the retina. And then caspase-3, we see predominantly in the photoreceptor layer, the rods and cones, there's a tremendous amount of apoptosis. This is the dark stain here for caspase-3. This is the cleaved form that's active in killing the cells, whereas we see none in this layer in wild-type mice. Now, in the RBP107 P10 mice that get retinoblastomas, we also see high level of proliferation in the retina. Now, this I'll need to expand on in the next slide. We needed to do more experiments to show that apoptosis is actually being suppressed by P10 deletion here, because you can see some regions still contain cell death, whereas some regions, these regions that were highly proliferative, are basically shut off for apoptosis. But this wasn't good enough. It didn't really make the point that we were trying to determine. That is, does P10 loss suppress RBE2F-dependent apoptosis in this context? And to do that, we needed to separate the cells from these populations. So the GFP, or the Cre that we were expressing in retinal progenitor cells, is fused to GFP, and some of the cells are positive for Cre, and some of the cells are negative. So what we really needed to do is separate out the Cre positive from the Cre negative based on the fusion to GFP. And what we found is if we took cells from these, separated into GFP positive cells, basically these express Cre and they've lost RB and P10. We see in the RBP107 mice a massive spike in the number of tunnel cells here that's diminished significantly in the P10 knockout. And all this apoptosis that we were seeing, it turns out to be GFP negative cells that had not expressed Cre recombinase. So what exactly is causing the cell death in these mice is not exactly clear, but probably because of some disorganization of the retina and or loss of access to various trophic factors, for example. But the summary here is that PTEN loss is suppressing RBE2F apoptosis in a cell-intrinsic manner. Now, if we extend this out to 30 days instead of 7 days, the phenotype is striking. So in these E2F activated mice, where we were seeing massive cell death, this is a close-up of the retina, which is basically destroyed compared to a normal retina. In other words, this latent, or this E2F apoptosis activity has virtually eliminated the retina here, whereas in the RBP107 PTEN mice, you see that high levels of Ki67 proliferation, apoptosis is basically shut off. It's consistent with the bilateral tumor phenotype that we're seeing in these mice. Jacob?
I don't even know whether mice have been able to gland, but do you see any tumors inside of the brain?
Yeah, we haven't spent a lot of time looking at that, but it metastasizes back through the optic nerve into the brain.
How about pineal gland? Because we sometimes see the trilateral.
Okay. Yeah. Yeah. That's not something that we've looked at. Yeah. Yeah. I would not be surprised. I mean, these tumors get so massive, so big, and it's in.
Very cool to show them.
Yeah.
Because we have the brain somewhere.
Yeah. Yeah. We've got plenty of samples, so we should look at that. Okay. So, and this is Kaplan-Meier analysis. So, not surprisingly, the RBP107 PTEN mice dramatically die quickly. The RBP107 mutant mice live fairly well out to a year, but then they pick up unilateral tumors that end up killing some of them, and the control mice basically don't get retinoblastomas or die. All right, so to move on to the next question, and this is the second concept. So the first concept that we wanted to address, and what I've been talking about, is does PTEN and AKT activation connect with E2F deregulation to promote tumorigenesis? And that's what we've been describing. But the real next question is, how does AKT interface to control the function of E2F1? Now, the simplest explanation would be, since AKT is a serine-threonine kinase, is if it directly phosphorylates E2F to affect its function. We think that's not the case. There's no good AKT phosphorylation sites in E2F. We think likely a different mechanism is that one of the AKT target genes, and some of them are shown here, may then control the function of E2F. So I'll be describing this for the rest of the talk. Now, AKT has a well-known role in pro-survival anti-apoptosis. It's also very important in glycolysis, so it has other targets, and just some of these are shown here. The approach that we took, since we had no idea which, if any of these, would be important for connecting to E2F, was to use an unbiased approach and target numerous of these AKT targets for shRNA degradation, and then ask, when we knock them down, which one of these knockdowns affects the ability of E2F1 to induce cell death? Now, the way that we chose to do this was to knock them down in a cell line that we have a lot of experience with. These are Ref52 cells, and a little bit of background with these. These are rat embryo fibroblasts, so it's an in vitro assay that we're using. We have a lot of evidence that we can ectopically express E2F1 in this background and cause the cells to undergo cell death. And this is partly by nature of our ability to deprive these cells of serum and shut off PI3 kinase AKT signaling. And by contrast, if we keep growth factors around, that was how we initially documented that PI3 kinase and AKT is able to suppress E2F apoptosis here. So we generated these cell lines in Ref52 cells and then asked if we ectopically express E2F1, so this isn't a control cell, and the assay here is measuring the percent cells that are positive for cleaved caspase 3 just by flow cytometry. So we see a spike in apoptosis by E2F1 in control cells. Now, the important point is that most of the things that we knock down here have no effect on E2F's ability to induce apoptosis. In other words, they're irrelevant for this phenotype. But we notice that when we knock down FOXO transcription factors. Here's one that just knocks down FOXO4 directly, and we have another siRNA or shRNA here that targets both FOXO1 and 3 for degradation. What we observe here is that E2F is no longer able to induce apoptosis, and it reverts back to control levels. So the idea then, and I won't show data for this just for the sake of time, but we have a lot of in vitro data where we've been able to show a direct interaction and a physical complex between E2F1 and FOXO transcription factors. And this is interesting for a number of reasons. So FOXOs are transcription factors that induce stress-responsive genes. They also induce pro-apoptotic genes. And you can imagine there exists sort of a Venn diagram of the genes induced by each of these factors, with some of them being pro-apoptotic. The other thing that we were able to show is that by CHIP, E2F1 and FOXO sit down on these pro-apoptotic genes to regulate the expression. So the model that we're going to be testing is to take this back in vivo into the mouse model and see if the PI3K AKT FOXO recapitulates what we were seeing with the PTEN knockout to drive retinoblastoma formation. An important point here, this interaction between AKT and FOXO is a direct phosphorylation target of AKT, and when it's phosphorylated, it is inactivated. So you see two contexts here. One, where AKT is not active, either, let's say, for example, one physiological context where this could happen is a cell where RB pathway was mutated and E2F levels go up, but there's no correlating pro-growth signal that would come through PI3 kinase or AKT. In this context, E2F levels would be able to interact with FOXO and induce expression of the pro-apoptotic gene. However, during normal growth or in a cancer that's activated PI3 kinase and AKT, by nature of this inactivation of FOXOs, what we've shown is that E2F is no longer able to transcriptionally upregulate these pro-apoptotic genes. But the next question then is, do these pathways, again, matter in vivo in the context of tumor formation? Now, to address this, we used in vivo electroporation in newborn mice in the mouse models that we had previously set up for retinoblastoma. So basically the concept is, can we electroporate in plasmids for the PI3 kinase, AKT, or FOXO pathway into newborn mouse retina? And we did this by subretinal plasmid injection. So these were actually injected into the subretinal space, and then we used in vivo electroporation using the system that you see here. First, tweezer trodes that contain a positive and negative node and couple over the mouse eyes and electroporate the negatively charged DNA towards the positive anode into the mouse retinal cells. And this is just an example of what we've done with GFP expression in normal newborn retina. So this is a whole eye here and a section of it. And you can see nice GFP expression in these sections from the electroporated mice. So then the question is, if we manipulate the PI3 kinase, AKT, or FOXO pathway, specifically in these mice that lose their retina by 30 days, are we able to see an induction of retinoblastoma? So there's kind of a lot here. I'll take you through this. We're using two different types of mice for this. First are just control mice, and the second are the DKO that activate E2F activity. So in the control mice, just putting in a luciferase plasmid and then imaging, we get nice plasmid expression in the newborn mouse. This is a control plasmid electroporated into control mice and harvested 60 days later, and a section of this is shown here. So we see normal retinal development. Now we see something odd when we transfect the rationale for using a dominant negative FOXO is because FOXOs, like E2Fs, are a family of transcription factors, and what people have shown in mice is that knockout of any single FOXO is basically not enough for any phenotype, and it requires knockout of FOXO1, FOXO3, and 4 in the mice to really get a phenotype. So we're working with a number of knockouts here. We decided instead to use a plasmid that knocks down the activity of all FOXOs. So this dominant negative FOXO is close to normal. It contains a normal DNA binding domain, so it can bind FOXO elements and the promoters of genes, but it lacks a transactivation domain, so it can't turn them on, and it can't express them, so it's dominant negative. We see that in normal control mice, just putting in the dominant negative FOXO has a dramatic effect on normal retinal architecture in that the photoreceptor cells wildly proliferate for a while and then stop. These mice never get tumors, but we see this routinely. So FOXO is having an effect in the normal mouse development of the retina. But here, kind of the key portion is down here. So you recall these mice, the CHX RBP107, these are the mice that by 30 days, you can see it here, lose the retina through E2F-dependent apoptosis. Now, if we put a control plasmid in here, this is what it looks like. The retina is still destroyed. However, if we use a dominant negative FOXO in this context, it's able to block this retinal destruction phenotype and cause retinoblastoma by 2 months. If we do the same thing with the constitutively active form of AKT, we see tumor formation, a retinoblastoma formation, and then this constitutively active form of PI3 kinase also is able to revert this retinal destruction phenotype and cause retinoblastoma. And mechanistically, we show in our final data slide here that this occurs through the suppression of RBE2F apoptosis in this context. So again, a control pre-GFP plasmid was transfected into these mice, and on its own, in the RBP107 mice, we see E2F-dependent apoptosis measured by tunnel-positive cells here. Activation of PI3 kinase or AKT or FOXO or dominant negative FOXO in this context suppresses the RBE2F apoptosis phenotype here, which correlates with the rapid tumor emergence in these mice. So again, to reiterate the model slide here, here we have a normal situation where RB is not mutated, PTEN is not mutated. In this situation, the phenotype is that E2F activity is low. These are not proliferating by nature of having RB around. There's baseline proliferation and baseline apoptosis. We don't see tumors in this situation. On the apoptosis genes that we've looked at, FOXO can be there, but E2F is not expressed, so they're not expressed. In a situation where you lose retinoblastoma through a mutation in the pathway, E2F levels quickly arise. And in this context, if PTEN is active and the PI3 kinase AKT pathway is basically not active in this context, the phenotype that we see is E2F and FOXO driving expression of apoptosis genes to suppress tumorigenesis, which is what we see at 30 days. There's no tumors in any of these mice. The retina is destroyed because of this E2F-induced apoptosis. However, then, if we activate AKT either through mutation of PTEN or through direct expression of an active form of AKT or PI3 kinase or suppressing FOXO function, apoptosis, again, is shut down in this context. Tumors, retinoblastomas quickly emerge, and E2F, even though it's around, it's sufficient to drive proliferation gene expression, but it can't induce apoptosis in this context. So I've got 2 slides here for future steps. What are some of the questions that we're asking now? One of them is looking at these early tumors that are forming. So these are the 3 genotypes that I've described to you. Control, RBP107, and RBP107 PTEN, which get the tumors by 30 days. And what we've done is harvested retinal cells from day 0 mice and day 7 mice, and we're asking, and we've gone through the procedure of isolating just the Cre-GFP positive cells because we showed from the apoptosis experiments that if we don't do that, there's a whole bunch of retinal cells that have not been affected by these knockdowns that would just clutter the data. So we're just isolating the RBP107-P10 deficient retinal cells here. And we're in the process now of getting data back for RNA-seq analysis or next-gen expression analysis. We think this is interesting because it's, to my knowledge, some of the earliest evidence and quickest formation of solid tumors. So the questions that we'll be asking are, what are some of the genes? Are developmental genes regulated in this context? Our expectation would be that E2F proliferative genes are through the roof in both of these contexts, but that apoptosis genes would be upregulated here, downregulated here, and then what else we see in this context. The second question leads into the next slide. The whole hypothesis, or one of the next steps and one of the rationales of what we're interested in here is the concept of if we administer a PI3 kinase inhibitor in this context to shut off AKT signaling, our expectation would be that we would be able to revert these tumor phenotypes back to this situation where E2F levels now are able to induce cell death because of the inactivation of FOXO. And that's something that we're directly testing now with BZ235. So the question then is a pilot study on functional consequences of PI3 kinase inhibition using this mouse model as a preclinical way to study the effects of PI3 kinase inhibition in this context. So this is the approach that we're starting with. This is to use not exactly the standard of care, but it is in a clinical trial, accruing clinical trial at Cincinnati Children's Hospital Medical Center with James Geller. So they have a pilot study of topotecan vincristine with subconjunctival carboplatin administration. And this is typically for patients with extreme bilateral retinoblastoma, either recurrent or refractory, so difficult tumors. The rationale for subconjunctival carboplatin is to avoid some of the myeloablative features of the systemic administration of these drugs. So the first step that we're trying with our mouse model is a carboplatin intravitreal. This is a little different from subconjunctival, and intravitreal administration of drugs has been controversial because of the fear of dissemination of tumor cells out of the vitreous. Retrospective studies that have been done recently really show that this risk is negligible. It's typically thought that this might be an approach in cases where vitreal seeding is a problem. So we're testing carboplatin intravitreally, topotecan intraperitoneally, and then BZ235 is a PI3 kinase mTOR dual inhibitor that's undergoing clinical trials for a variety of different cancers by oral gavage. So testing carboplatin and topotecan at day 1 with 5 days of oral gavage with BZ235, waiting until the end of 3 weeks and repeating this for 3 regimens. The questions that we'll be asking then are, do we see induction of E2F-dependent cell death by administering? Do we see a regression of tumors? And then Kaplan-Meier analysis for survival. And then finally, we're also interested in the effects that BZ235 or PI3 kinase inhibition has on gene expression and fitting this into the RNA-seq data that we're getting back with, again, the expectation that we'll be affecting E2F's ability to induce apoptosis. And then I'd like to thank people in my lab, shown here, collaborators. Aaron Sarver at the university here is involved in the bioinformatics projects. Mike Dyer is a retinoblastoma person at St. Jude that we've interacted with, and Rod Bremner is retinoblastoma at Toronto. Grant support, NCI and American Heart Awards for this project. So, be happy to take any questions.
In your mouse model, do you see
retinoblastoma early on, sarcomas, then bladder cancers, lung cancers, and things. Do the mice just not live long enough, you think, or?
There's two questions. The short answer is no, they don't develop other cancers. And I think partly the reason is because we're using this retinal-specific Cre recombinase. So it's just really affecting the retinal cells and knocking it out there. I'll tell you just a short story. Actually, it's probably not a short story, but we haven't really been able to do this the way that you've described. We tested this, actually, the same concept in osteoprogenitor cells with thought, since RB is frequently mutated in human osteosarcoma, one of the main ones, would we see osteosarcoma development? And actually we saw, maybe in retrospect, not a surprising phenotype, but rather than osteosarcoma formation in this context, we actually saw de-differentiation down the adipocytic lineage. So it really is not, I think the short answer is we need to use a tissue-specific Cre to ask the question that you're addressing, since these are lethal. And it's not just affecting the E2F apoptosis induction, but it's also affecting RB's ability to promote differentiation down different lineages.
The adipocytic development is interesting because lipomas are a hallmark of some families with constitutional RB, just widespread lipomas.
Yeah.
So it's interesting that you're seeing the same thing.
I think conceptually it's very interesting from a paper standpoint, not as interesting as if we had seen osteosarcoma.
Right. And then just my last question is, one of the hallmarks of retinoblastoma is it stops, right? So up to, very, very rarely ever see this occur after 6 years of age. And even in a patient with a heritable RB who's got plenty of precursor mutations waiting, it stops. Is there any data on what actually changes at a cellular level to stop it from continuing at 8 years, 10 years, 15 years?
I think it's complicated. The fact that it only happens during retinal development, I think really suggests that a mutation in RB, or let's say the second hit in RB, really needs to occur in a very discrete cell population at a very discrete time to take off into a full-blown retinoblastoma. Something that I didn't get into here, some of the more recent data from humans have suggested that perhaps cones are the cell of origin. People have mutated RB in embryonic cones and actually, without other mutations, can transplant those back into nude mice. They give rise to retinoblastomas, whereas the same type of experiment in other cells of origin, other retinal cells, doesn't do the same thing. So it may be that it needs to happen in a very specific progenitor stage. The other thought that I didn't really delve into is, what's suppressing apoptosis in this context? There's some evidence, certainly other mutations arise, and some of those affect the ability of the cell to undergo apoptosis. There's a whole other conceptual approach that maybe this is like some of the other pediatric cancers, medulloblastoma, for example, that perhaps there's something intrinsic in the cell of origin that causes it to be resistant to apoptosis, and that may be through certain cellular pathways that are just innately strong in those particular cells, whereas if you knock out RB in a later stage, maybe the cells just immediately die. If you knock them out in this certain progenitor cell that's innately resistant to apoptosis, and they take off into a full-blown cancer.
So this relates in part to your question, Joe. But the first question I have is that how frequently do mutations in the RBE2F pathway, are they acquired in other cancers?
The textbook view now is that the RB pathway is mutated in all solid tumors. RB mutation itself is fairly rare. A lot of times you'll see upregulation of the cyclins that end up phosphorylating and inactivating RB, but more commonly you see mutations in the cyclin. The CDK inhibitors like INK, for example, is more widespread. What that means from why RB, why a CDK inhibitor, probably I think it's still very poorly understood. why RB in retinoblastoma and osteosarcoma versus the CDK inhibitors in other contexts? And it may be that some discrete function of RB is critically important for the development of retinoblastoma and osteosarcoma. One thing that's, if you mutate a CDK inhibitor, basically the effect is that E2F activity is through the roof. But you still have RB around, and maybe it's doing other things to promote differentiation or control some other aspects of tissue homeostasis or survival. But in an RB mutation, both functions are gone. The ability to suppress E2F, but also the ability to differentiate down specific lineages. So I think it probably has something to do with terminal differentiation of retinal cells.
Joe, does the timing of the sarcomas occur normally? I mean, so even though we have the known RB mutation, but their osteosarcomas are occurring at the appropriate age range? Any other questions? Well, thank you, Tim. It was great. Thanks.